Hi everyone, this is Matt from AB Studio Inc. I would like to share with you how a non-human antibody is humanized at AB Studio. Before I introduce the antibody humanization process, I would like to briefly introduce who we are and how we do our work. AB Studio is a biotech platform company supporting novel and complex therapeutic antibody development. We combine computational design with traditional antibody discovery and engineering technologies to develop novel and complex therapeutic antibodies. So here's our unique antibody humanization process. Before I introduce the antibody humanization process at AB Studio, first I want to explain the concept of antibody humanization. When we ask ChatGPT, what is antibody humanization, here is ChatGPT's answer. Antibody humanization is a process of modifying an antibody that is originally sourced from a non-human species, such as a mouse or a rabbit, to make it more compatible with the human immune system. Since non-human antibodies can trigger an immune response in humans, humanization involves engineering the antibody to resemble more closely the structure and sequence of human antibodies while maintaining the specificity and affinity for the target antigen. This is typically done by identifying the parts of the non-human antibody that contribute to its immunogenicity and replacing them with corresponding human sequences or modifying them to minimize their immune recognition. The resulting humanized antibody is less likely to elicit an immune response when administered to humans, which is an important consideration for therapeutic applications. Antibody humanization is a crucial step in the development of many therapeutic antibodies, particularly those used to treat cancer, autoimmune diseases, and infectious diseases. So, if we say this concept is correct, then the next question is this sufficient? Based on the concept of engineering the antibody to resemble more closely the structure and sequence of human antibodies while maintaining the specificity and affinity for the target antigen, many humanization service providers guarantee the following two features post-humanization. The first feature is humanization percentage within an acceptable range. The second feature is binding affinity reduction, if there is any, to no less than 50%, that is, no more than twice the KD, compared to that of the parental antibody. Antibody humanization at AB Studio has a new standard. This is humanization with safety, efficacy, and manufacturability balance. As a result, we can guarantee this new feature other than the two features most other service providers can guarantee. We also guarantee two more features. These are, number 3, if there's any increase in aggregation propensity, it will be less than 10% compared to that of the parental antibody, as measured by DLS. And number 4, if there's any reduction in thermostability, it will be less than 10% compared to that of the parental antibody, as measured by DSF. Our overall research goal is to guarantee a humanized antibody with safety, efficacy, and manufacturability. This is the workflow of antibody humanization and optimization at AB Studio. Once we receive a non-human antibody sequence, we will first build up a 3D model of the FV of this antibody, and also we will input this FV into a database search for human germline antibody sequences, which share high homology to that of the query non-human antibody. Then we select the top leader human germline acceptor sequence, perform CDR grafting by replacing the CDRs of the human germline acceptor sequence with those of the query non-human antibody. Then we will analyze how many T cell epitopes, B cell epitopes, MHC epitopes, and antigenicity epitopes there are on the 3D structure of the non-human FV, and then also analyze how much aggregation surface, how many positive patches, negative patches, hydrophobic patches, and post-translational motifs there are on the 3D model of the FV structure from the parental antibody. The design process will be attempting to remove immunogenicity epitopes or minimize immunogenicity epitopes while also reducing aggregation surface, positive patches, negative patches, and attempting to remove post-translational motifs. After design, usually we will create more than one variant. Sometimes we will design something like 3 heavy chains and 4 light chains, and then there will be 12 total combinations. Then we will first perform in silico analysis to determine the difference between the parental sequence and the humanized sequence for the 3D information, including change in aggregation surface, positive patch change, immunogenicity epitope change, and post-translational motif change. Change means, for example, how many aggregation surfaces are there on the new humanized FV structure compared to the parental FV structure. If the numbers of aggregation surfaces are similar, then what about their sizes? Did we increase the size of an aggregation surface or not? The increase in size or number of aggregation surfaces would be bad features for manufacturability and should be avoided. Then, after we balance all these features and finalize our design, we will test our designed antibody humanization candidates on the bench. The first round is to screen for binding affinity or specificity, and in the second round we screen for manufacturability. For antibody humanization design, usually modeling of the antibody FV structure is sufficient to guide the design. Sometimes we'll also make an antibody-antigen complex structure. If the complex structure matches certain criteria to enable us to think it's accurate, we will also use the antibody-antigen complex structure to guide our humanization design. So, for example, this is a model structure of an antibody-antigen complex. The green part is the FV of a query antibody, the cyan part is the antigen. Pink residues are the antibody residues that interact with the antigen, and the yellow residues are antigen residues that interact with the antibody. So by analyzing this antibody-antigen interface, we can better humanize the antibody without loss of affinity. This is step 2. After a model is built up, we will mark antigenicity epitopes on the FV model. We have licensed software to predict T-cell epitopes, B-cell epitopes, MHC epitopes, and antigenicity epitopes. Step 3 is surface analysis. By using licensed software, we can analyze how many positive patches, negative patches, and hydrophobic patches there are on the 3D FV model, how many aggregation surfaces there are, and how big each aggregation surface is on the 3D FV model structure. Step 4 is 3D PTM, or post-translational motif analysis. We can analyze this based on the 3D FV structure to determine how many post-translational motifs there are on the 3D FV model. What is the side chain exposure percentage? If a motif is exposed on the 3D surface, it has high risk. If it's buried, it may be solvent inaccessible and has low risk. Step 5 is antibody humanization design. So the rule for design is, after grafting a query antibody's CDR onto a human germline antibodies framework, we try to reduce back mutations. Back mutations means changing human residues back to non-human. So in what situations are back mutations needed? We summarize this as the following. First, if replacement of a human residue leads to loss or gain of an ionic bond, a hydrogen bond, or a hydrophobic interaction with a CDR residue, a canonical residue, or a VHVL interface residue, or a Vernier zone residue, back mutation is needed. Secondly, if replacement of a human residue leads to an increase of antigenicity epitopes, aggregation surfaces, positive patches, 3D PTM, or a significant change in 3D PI, back mutations are usually needed. Other than following these rules to humanize the antibody, during the antibody humanization process, we also do some optimization. The optimization needs to take into account a lot of balances. For example, this is a paper published in 2010. The author found that in the CDR domain of a certain antibody, there is an N-glycosylation site. They mutated the N to a D, so the N-glycosylation is removed. However, the new protein, though the affinity is similar to that of the parental antibody, is prone to aggregation. Later on, they performed detailed studies and they found the reason for the N to D mutation causing aggregation is when the N-glycosylation is there, the glycan forms a kind of umbrella that covers the aggregation surface. Once this umbrella is removed by mutating N to D, getting rid of glycosylation, the aggregation surface is exposed. As a result, the protein aggregates. So this is a typical example to show a balance between heterogeneity and aggregation. If you maintain N-glycosylation, the antibody would have heterogeneity downstream in CMC. But if you remove the N-glycosylation, the antibody would aggregate. So this is a feature during the design process that needs to be balanced. Here's another example. So DD is a typical isomerization site. The question is, should we remove DD in all situations? The answer is no. We had a case study of this before. In that case study, there was a DD isomerization motif in the CDR domain. However, both of these aspartates are involved in ionic bond formation. As a result, the carboxylic acid moiety, which is pointed to by the red arrow, now is not available for isomerization, and there is no need to remove the DD. So, the summary of my presentation is, we think the humanization of antibodies is an art of balance that includes balance of humanization optimization and also includes balance of different kinds of optimization. This is a review paper published by AB Studio in 2021 talking about optimization of therapeutic antibodies via computer-aided design. So for anyone who has interest and wants to learn more details about antibody humanization and optimization, please feel free to request the PDF link of this paper from us. Thank you very much. If you have any questions or concerns, please feel free to contact us at abstudio.info@antibodystudio.com. Thank you again. Have a nice day.